you all for coming. And you've heard this before. Normally, Peter Bergstrom is the person who gives the State of the Magothy address, but Peter is actually fully retired now, and he's in Vermont. So I had the opportunity to put together the State of the Magothy this year, and so I will be presenting it to you. So, yeah, this is pretty much how the river looks these days. And weren't there some great shots from that video? I just love that video. It makes me remember summer's coming, and we will be out on the river someday. So, the Magothy River Index is made up of 3 parts. It's an opportunity for us to crunch the data that's been collected by our volunteers and some here from the community college over the year. And the 3 parts consist of the coverage of the SAV, the submerged aquatic vegetation. Of course, we would like to have these types of plants. Here's a picture, you know, if we have good plant growth, you see it sort of at the edge, at the surface of the water, blooming. It provides food and habitat for fish and shellfish. It's very important habitat for the river. Water clarity, we measure with a Secchi disc. You know, we drop it down and we see how far it can go into the water before we lose sight of it. And what we do is we accumulate over the course of the summer. We use this Secchi disc at all of our sites, and every time it's at 1 meter or greater, that is a passing grade. And the idea for that is that we need to have water penetrating 1 meter into the water in order for the submerged aquatic vegetation to grow. So that is how much light we need. And then the third item is the concentration of dissolved oxygen. So we measure that at depth in the deep stations and at surface in the very shallow stations. And again, every time we have a DO value greater than or equal to 5 milligrams per liter, that's a passing grade. So the idea here is that we need about 5 milligrams per liter for good survival of fish and crabs. So those are our 3 parameters that we use to calculate the index. And so, as I said, we get the data from VIMS for the SAV area. Although this year, VIMS has not published the data for the Magothy yet. I did try to have a little chat with them, and they think they may have seen a little bit, but we have no quantitative data from them. The water clarity with the Secchi disc, here are 2 of our volunteers here. It looks like that picture kind of got a little bit squished down, but here's our 2 of our volunteers who are out there on the river doing some sampling. So dissolved oxygen we measure with a meter. Water clarity we measure with the Secchi disc. And then the letter grade is based on the average of the 3 values. So if we have, you know, great, if we get, you know, 100% of what we'd like to see, that would be an A, 60 to 80 is a B, 40 to 60 C, and the 20 to 40 is a D. So we are actually in this 20 to 40 range right now. Anything less than that would be an F. So here are the numbers, and you have this figure in your newsletter. Here are the values that we got for this summer, or this year, compared to, we've been doing this index since 2006. So what we can see here, so this blue line is our dissolved oxygen concentration. And the good news here is that we have the best dissolved oxygen in the water column. This is both creeks and main stem of the river that we've ever had since we've started doing this in 2006. And so good dissolved oxygen levels. Our water clarity is this lighter blue line, and it was, you know, much better in 2006. This was when we had the dark false mussels the last time. Lots of filtering going on by the mussels and pretty good water clarity at that time. Once the mussels died off, we had a steep decline in the in our water clarity, and it has been, you know, quite low, although it came up a little bit last year and a little bit more this year. So we did see somewhat increase in water clarity. Again, the mussels are back, and we think that has a lot to do with the return in water clarity. Submerged aquatic vegetation, we had to map it here as no SAV because VIMS thinks we didn't have any. But I just learned tonight from this poster right over there in that corner that the county has been having Bayland, this consulting firm, map SAV in the river. And actually, they have mapped over 70 acres of SAV in the river. If I had had that number when I did this calculation for our index, I think I would have had it come up to at least 40%. So maybe we'll be able to do a little bit of addendum, or maybe for next year we'll hope to see it up higher because we'll be able to have some better SAV data. But the good news is that this is the overall, this is the average of the three now. So for this year, we're at 32%, and we were at 22 last year. So it actually is a substantial improvement. And the one which is really significant, statistically significant, is this increase in oxygen. So that's the good news. It's an improvement. It still is a D, but it's better than a D minus, which is what we had last year. So if we break down the index into the various parts, these are all the sites where we sampled this last year. Again, this has been stretched out. This kind of weird looking picture of the river, but I guess we'll get used to it. So we have main stem sites here. In this area here, plus this one in Tar Cove. These are our main stem sites. There are 9 of them, and then there are 9. We're calling this one up here in Beechwood, even though it is the main stem of the river, we're calling it a stream. Well, you could call it a main stem or a stream site, either way. It wouldn't matter in terms of the calculation. And then these are all the creeks here where we have data. And the people who have been doing the monitoring for us, Chris Kirshner did the monitoring in Deep Creek and in Little Magothy. The Lamans who were here, and Steve Troy and Dave Kemp, they did the monitoring for Cypress. I think they're all here today, right? They did Cypress, Dividing Creek, Mill Creek, and this Olmsted Point. And then all the rest of the open water by boat and all of these ones up here and this one on the North Shore, those were all monitored by Paul Spadaro. So we have a variety of people who were working on this. And my students and I work on the Mill and Dividing Creek also in the summer, so some of that data came from us.
Clarity or oxygen?
So what this is a combination. This is the index for the combination of the three. So what we can see here, the green ones here are telling us this is our best water quality then, 40 to 45. This would be a grade of a C. And if we only had the main stem sites, we would actually have been a C this year. So the green ones here, and even all the way up here to Beechwood, this is right by Beechwood Park, you know, our newest park on the Magothy. And water quality was very good up there. So the green ones are the best water quality, combination of SAV, which is 0, oxygen, and dissolved oxygen and water clarity. So the blue comes next, so we can see the headwater, or the part of Old Man Creek that's closest to the main stem is blue. Tar Cove is blue, and this is MR6. This is the one site that the state always monitors. This is where DNR goes. They consider this to be typical of the Magothy. They only do one site in the river, and that's it, MR6. So that is in blue. And then, so that's, not quite as good, but still not bad. Then the yellows, we're getting down into a lower rating there, and these would be these 3 over here, plus the mid-Cattail Creek point. So the reason why these 3 are lower for open water sites than the others is poor water clarity. Dissolved oxygen levels were good here. This is an oyster reef, Dobbins Hill Reef. Rock Point is a reef, and this is the sailing mark out there, F2. These are, the dissolved oxygen was good, but the water clarity was bad. That's why they end up in yellow. Same thing over here for cattail. And then we didn't have any in the orange, actually. And then in the red, we have these 3 creeks over here, Cypress, Dividing, and Mill Creek. They were lower in both dissolved oxygen and in water clarity, so they end up with this, value that is right on, 20 is the edge of what a D would be. And then Deep Creek and Little Magothy, and then way up here in the headwaters of Cattail, these 3 came out. Actually, they are in the F range, right? F starts at 20. Or 19, actually. So we can see these are the areas where we really need to, I think, concentrate on for doing some improvement on these creeks. So that means they had low dissolved oxygen and low water clarity. All right, so let's see. Just to remind you of where we used to have a lot of SAV, this was in 2005, and all the red all along the shoreline here, this is where the aerial surveys had shown the presence of SAV in the fall each year. 2005, we had 308 acres. Now here we are over here in 2010, 5 years later, only 2 small sites here with less than 2 acres. And then from 2012 to 2014, according to VIMS, we have no SAV. But we know, according to more detailed surveys done with kayaks and small boats, that we do have some SAV. So it's a little bit of a mixed story there on the SAV. So just to show you the long-term trends that we have for the main stem site. So these are the ones out in the open water. for our dissolved oxygen going back to 2002. We've been monitoring these sites all the time. And again, you can see this is dissolved oxygen and from 2014, significantly increased. This is a statistically significant increase in DO for between 13 and 14. The MR6 is always a little lower than the others because it is the deepest point, and so we have a site that is down as far as, I think, 3 meters. So it's really our deepest site that we monitor. So the oxygen levels are a little bit lower there. So it's kind of ironic that this is the site, of course, that the state uses. You know, if they'd gone to any of the other sites, they would have much better values, but this is the site that the state uses. So that's the trend that we see for dissolved oxygen in our main stem sites. And over here, the same sites for water clarity. So what we can see is we have kind of a mixed story in 2014. Some sites show improved water clarity, some sites show decreased water clarity. So we don't have a significant increase there, but we do in dissolved oxygen. All right, so what I want you to notice here in water clarity, again, here's 2004. This is when we had mussels, and right in this area here, and that's also when we had the good SAV in that area right there. All right, so that's what we have for the index. But we also have some other things going on that we wanted to talk to you about, and one of them that we started this year, 2014, because we had the poor SAV reports from the VIMS, from the aerial surveys, we said, you know, we know there's SAV in the creeks. We've seen it. We've all seen it. Those of us who are out in the creeks, we know it's there, at least early season SAV, the horned pondweed primarily. So we got a program going, and Jeff Reagan agreed to coordinate this program. So we had a little session at the college where we kind of talked about how we would go out, and we would have kayakers go out during the course of the summer and go up into these small creeks and try to see if they could find any SAV, identify it, and also try to see if we could get it a little bit of a quantitative value for these. So all of these red points that we see here, these are all areas where SAV has been found. The 3 yellow points are launch sites for kayaks, just sort of for a reference there. So some of these, this is a map on Google, and if you click on those points when you go to that map, some of them will say, you know, dense, lush growth of SAV. Others will say very sparse. So we do have some of that little bit of quantitative data, but we don't have good areas on a lot of them. So we couldn't use this in our index yet, but you know, we're hoping that as time goes by, we can perhaps try to get a better handle on making this quantitative. Now this is mostly early season grass. It's this horned pondweed. This is what it looks like. And it's pretty much done by mid-June. But it still is an important fish and waterfowl food source habitat. And it is still an important grass, even though it's not seen by aerial survey in September. So we're hoping to continue with this year, and Jeff Reagan is right back there, right? And he's going to be taking sign-ups, right? And we're going to be doing it again this coming year. So if you want to go out, it's really fun. You pick a nice day. A lot of these points were picked, at least when I did a lot of them over Memorial Day weekend. And we went up to a couple of these sites here, and get to go out on a nice day and look for grasses. It's fun. So there's our SAV hunt. We also, I want to talk to you a little bit about the bacterial water quality monitoring that we do. This is the communities sign up to be monitored. And I'm passing over this program this year to Tammy Demansky, who is sitting back over there. Tammy? She will be running this program this coming summer. And so this is the organism that we monitor for. It's called Enterococcus faecalis. This is an organism similar to coliforms. You may be more familiar with the idea of monitoring for coliforms. And, but the Enterococcus is similar in the sense that its native habitat is the gut of warm-blooded animals. That's fish and, birds and mammals, not fish, birds and mammals. This organism itself is not one that's likely to make you sick. But it tells us that if it's present in the water, there has been recent fecal contamination of the water. We don't know what the source is, whether it's human or not, we don't know. But if it's there, we know that water has been contaminated, and if the level is high enough of these indicator organisms, then water is considered not suitable for swimming. So we monitor water quality in communities that sign up. So we have a sign-up sheet. Tammy has brought some today. I think they're out front. And so communities sign up to be sampled either every week or every other week in the summer, just to see if their water off their piers or off their beaches is suitable for recreation. So we sample on Wednesday mornings, as you can see Tammy doing here. Then we bring the samples into the lab at the Environmental Center here on campus. And we filter the water samples so that we have a filter in here that will trap the bacteria on the surface of the filter. And then we take that filter and we place it on media and we grow it for a day. So this is a little petri dish with some agar in it for media to grow the bacteria. And then in 24 hours, each of the individual cells on this filter has grown into a small colony. And then we count the colonies, and that, because we know how much we filtered, that tells us how many of these bacteria were in the water. So we call them colony-forming units per 100 milliliter. That's the standard way of describing these results. And you can see why, because these are the colonies that we're counting. So that's what we do. So here are the data for 2014. We had 15 sites this year. This is the most we've ever had. And so what I've done here is I've tried to kind of crunch the numbers down so that we can look at kind of trends that we see at the different stations. But one of the things that we've always known is that after a heavy rain, we always have higher counts of bacteria. So that, you know, if we only sampled in a particular site only after a rain, we would probably get a lot of high counts. But what we do is we sample every Wednesday or every other Wednesday, so sometimes we get a rain event and sometimes we don't. And so that's why what I've done here is I've shown the data here for these stations both without the rain and those including the rain. So for instance, now some of these though, like here's Beechwood, there's the one that we have the site on for water quality monitoring, and it's a private house up near Beechwood Park. Notice there's no yellow line on here, and that's because they were being sampled every other week, and it just so happened that we never sampled them after the rain. So the one all dates and without rain would have looked the same. So rather than put that confusing result there, I just showed without the rain, this is what it looked like. So that's true for Beechwood, Fair Oaks, Cypress, Bayberry, and Deep Creek. Those happen to be the ones we were sampling on the alternate weeks, and they just happen to be ones that never were sampled within 48 hours of a rain. So, what I've expressed here, the data are as the geometric mean. So basically what this is a way to smooth out the extremely variable data that bacteria give us. Because bacteria can be hugely, very, very high one week and then lower the following week, we calculate this geometric mean to give a more kind of a smoothed value that is easier to interpret. And when we use the geometric mean for our scale here, 35 is the cutoff for safe swimming. So this dotted line is across here, and we can see that most of our sites then, with the exception of Dividing Creek at Manhattan Beach, most of our sites for without rain are good. You know, the water quality is good for recreational use. What's interesting here are to note Conkey Creek, and this is the Manhattan Beach side of Cypress Creek, and then Forked Creek. These are the three that are really showing high counts after rain events. So again, we can use these data in two different ways. We can say, is the water fit for swimming today? And we do post the data every week, so people who are at these sites can check their data every week. So it tells us, is the water good for swimming right now? But it also gives us this long-term trend. So we can say, are there sites here where we should be paying attention and trying to retrofit? And it looks like these are three sites here, Conkey Creek, this part of Cypress, and this part of Forked Creek that maybe could use some stormwater intervention because we're getting very high counts after the rain. So Tammy, as I said, has some applications here. Yes?
Just a quick question.
Yes.
Cypress and Cypress MB.
Yeah, so we have two sides of Cypress Creek there. The Cypress MB is the Manhattan Beach side of Cypress Creek, and the other Cypress is the other side of Cypress Creek. So that's the Severna Park side of Cypress Creek. So I did them on alternate weeks. One week it would be the Severna Park side. Well, I guess they're both Severna Park, aren't they? One is the side that you get to from Ritchie Highway directly. The other one is the one you get through from Manhattan Beach. So those are the two sides of Cypress Creek. Yeah. Same thing over here with Dividing Creek. We have Dividing Creek that you get to from Peninsula Farm Road, and then you have a Dividing Creek that you get to from Manhattan Beach. So there's two sides of Dividing Creek there. So we sample those on alternate weeks so that we would get best coverage. So that's what we saw for 2014. Now if we want to compare, yes? Oh, well, I'm so glad you mentioned that. Now here's Mill Creek. This is Mill Creek. It's right across from the community college. And we did have a spill in Mill Creek. On May 27th, there was that 300,000 gallon spill of sewage. So what I'd like you to notice here is that despite that, actually Mill Creek was in pretty good shape. And so what this tells us, it's actually in pretty good shape and not so good shape after the rain. But what this really tells us is that even though you can have a major sewage spill here, it's not, the effect of a sewage spill is not as impactful as the effect of the rain. So I think that's a really important take-home lesson for us all. You know, we worry about these spills, and they are problematic because, of course, it's not just bacteria, it's also nutrients that come in. But the effect of the rain on Mill Creek is actually more significant than the effect of that 300,000 gallon spill. I think that's sort of a take-home lesson for us. Okay. So I wanted to just compare. There were 10 sites that we did last summer that we did in 2013 that we also did in 2014. So I don't have a lot of comparable data here, but I just wanted to see, is it better or worse between 13 and 14? And the answer is it's been about the same. Again, the all dates and the without rain dates. So it was a little bit lower here, but that's not a significant difference between 13 and 14. And the without rain dates are basically the same. So I think we would say that the effect of stormwater runoff and water quality for swimming purposes, it was about the same between last year and this year. Okay, now we're also continuing with our Beechwood Park, and I won't really talk about that at all except to say that it is ongoing. And we've had lots of cleanups there, and we've had nature trails established there that have been mulched, and there's always a call out for more work to be done on that. So if you are interested in being up in that Beechwood Park area and helping out, we often have a call for needing help for that. We're trying to get a boat launch site put in there, so it would be a kayak canoe launching site. We're working on that. And then finally, the dark false mussels. I did want to talk about them a little bit because they did return. We think that they did help us with water quality. And so this is what they look like. If you haven't seen them, this is our ruler here. So this would be 1 inch on the ruler. And you can see these guys are generally maybe about 1/3 of an inch in length. They're quite small, but they can be very abundant. Here they are clustered on an oyster shell. So you can kind of get the relative size of all these mussels on a single oyster shell. And we learned a little bit about their natural history this summer. Steve Troy put out this set of tiles here. So this would be, this one here is the, let's see, get the right end here. This is this is 1 meter, right? This is 1 foot. 2 feet, 3 feet. No, that's the other way, isn't it? It's this way. 1 foot.
You were right the first time.
I was right the first time. This is 1 foot here. Okay, sorry. 1 foot, 2 foot, 3 feet. We're getting a lot of barnacles here. 4 feet, kind of a mix of barnacles and mussels. And then at 5 feet and 6 feet, that's where we get a really good growth of the mussels. So based on this, he just hung this from his pier and watched it over the course of the summer. So based on this little experiment here, we can say that the mussels do seem to do best at 4 to 6 feet. So it might be that a lot of us might have mussels at our piers, but we don't know about it because it's down at 4 to 6 feet and you can't see them. So that's one thing that we learned. We also learned that they reproduced at least twice in the course of the 2014, between May and September. We had at least 2 generations and maybe even 3, depends on how you interpret the data. So that's how we got such a big biomass produced. And so finally, we did want to just talk a little bit about the situation with the dogs that have gotten sick, and even some dogs have died as a result of eating the mussels. And we do have that brochure out there and a poster that we would like to put out, and hopefully it'll go at some of the parks. So it turns out what happens is that dogs really like to eat these mussels. Now we've also seen crabs eat them, we've seen waterfowl eat them, and we haven't noticed any ill effects from that. But dogs seem to find them delicious. And that's a problem because they have caused liver failure in dogs. And some dogs have died after eating them. Some have been able to be treated and have been brought back. So we have that brochure out to talk about it. We worked with Dr. Carl Rogie, who is the veterinarian from Severna Park. And so we put together that little brochure, that little one-pager there. If you want to take a look at it, it tells you basically something about what we think causes the toxicity of these mussels. It's either algae that produce toxins that the mussels concentrate. You know, these mussels are filter feeders, so they concentrate whatever is in the water column. And if they concentrate the toxins of the algae, then that might be what makes them toxic. Or it might be that the mussels, once they are grown on here and once they die off, that they start being decomposed by bacteria, and maybe the bacteria that are decomposing the mussels are producing toxins that are making them sick. We really don't know. We're trying to learn, and hopefully we're going to find more about this soon. But all we know right now is that the mussels are toxic, and we're really recommending that you really be sure to keep dogs away from the mussels. If they do eat them, you know, watch them really closely, and if they appear sick at all, call your vet right away. Yes? Oh, great question. I think it's because of salinity. I think that's the primary answer here. We've had a pretty wet year last year, and this year we're pretty wet, and our salinities are down. And these are basically kind of very low salinity mussels. Like when we had them in 2004, they came right after Isabel, when we had all that fresh water coming in from Isabel. And then they stuck around for a few years. So they seem to reproduce when we have the lower salinities, and then they stick around for a few years and probably stop reproducing once it gets saltier again. So if we had another wet summer next summer, we might see them doing even better. If it turns out to be a dry summer, we might not see them reproduce. But that's what it seems to me. It's primarily a salinity connection. Yes.
My jet ski floats,
Yes.
floats,
Yes.
pretty much
Covered.
put down there.
Covered. Exactly. And that's one of the things that I put in the newsletter. You know, these little floating water gardens that we have out there. We brought the one in from the Troy's house, brought it into the lab, scraped off the mussels. So this is only a float, you know, so by so. And we weighed the mussels that we scraped off it. 75 pounds of mussels came off a single float. So we're talking a lot of mussels here. And some of the dogs that have gotten sick have eaten mussels that have been scraped off. Like I just talked to one of my neighbors recently, and I didn't know this until the other day, but there were mussels that were on the prop from her boat had been pulled, and she just had some mussels on her prop. They scraped the prop. They were in her backyard. The dog ate the mussels in the backyard and got really, really sick. So you do want to be careful about that. Okay, I think I've gone way over my time budget, my time frame here, so I'm going to go real quick to say that we're also doing the floating gardens, and we're going to, Providence Center is going to, I think, going to be selling them this year. So we've got some sign-up lists for that. Do we have a sign-up for that, Paul? I think we do. And so we're going to continue with those. Those of you who have done it, you might want to go for another year. We have a couple different designs, so we'll see what will be the design for 2015. And so again, I want to just thank, we have a lot of, you know, we're doing a lot of projects, and we have a lot of really dedicated volunteers, and we just really appreciate everybody's help. And particularly I wanted to mention also the Anne Arundel County Department of Public Works Watershed Protection and Restoration Program, because they have really noted places in the Magothy where stormwater is an issue. And they have, like the poster over there for the Haskell Drive situation, they have come in and put in some stormwater retrofits. This is your stormwater fee at work, and it really is helping to improve water quality. And maybe some of the increase in water quality is due to these projects. Hard to know, but they must be doing something. Okay, thank you. Thank you.